Topoisomerase-based cloning consists on using the molecule topoisomerase 1, which recognizes a specific DNA sequence, then unwinds it and reattaches it again by the phosphate 3' thymidine. The PCR 2.1 topo vector can be used for direct insertion of PCR-amplified products with Taq polymerase in a plasmid vector for subcloning. For topo cloning and transformation into chemically competent bacteria, adding sodium chloride and magnesium chloride to a final concentration of 200 millimoles sodium chloride and 10 millimole magnesium chloride in the topo cloning reaction. For transformation of electrocompetent bacteria, salt must be included in the topo cloning reaction, but the amount of salt must be reduced to 15 millimole sodium chloride and 2.5 millimole magnesium chloride in order to prevent arcing. The PCR product must have a thymidine 3' outstanding that can be used to complement the protruding adenine PCR product, thus allowing the ligation of the PCR product into the plasmid vector. Now, XGOL is used to visualize cells containing recombinant DNA that have not caught a vector with the gene. If beta-galactosidase is produced, then XGOL is hydrolyzed to form 5-bromo-4-chloroindoxyl, which spontaneously dimerizes to produce an insoluble blue pigment called 5'-4'-dibromo-4,4'-dichloroindigo. The colony is formed by non-recombinant cells, therefore appear blue in color while the recombinant ones appear white. The desired recombinant colonies can be easily picked and cultured. Now, the PCR 2.1 topo has several elements important to work. These elements include the LAC-Z-alpha, which codifies beta-galactosidase, multiple cloning sites or polylinker, which is a short segment of DNA which consists of many reaction sites commonly used during procedures involving molecular cloning or subcloning. The T7 promoter, which catalyzes the formation of RNA in the 5'-3' direction. F1, which enables single standard replication and packing into phage particles. Canamicin resistance, which is a string of DNA that allows the organism to produce a protein conferring resistance to the common antibiotic canamicin. Ampicillin resistance, which confers ampicillin resistance to the cell that has the plasmid carrying DMPR gene. And PUC origin for sequencing inserted cloning into PUC plasmids.
The results include 20 white, round, smooth and regular colonies without any blue colony. According to the observations, the efficiency is 4 to 10 to the third colony-forming units per microgram of PCR 2.1 topo. Topo cloning is a method developed to ligate PCR products in about 5 minutes right after the PCR has been run. PCR topo contain a topoisomerase covalently attached by its tyrosine residue 274 in each 3' end of the linearized plasmid. It is necessary that a polymerase cleaves adenines in the 3' extreme to be able to interact with the thymines in the 3' ends of the plasmid. It contains two resistance genes to antibiotics, ampicillin and canamicin for the selection with E. coli. It contains echo R1 restriction sites flanking the insertion site, LAC operon promoter for induced expression and blue-white screening for selection of recombinants, F1 and PUC oris for plasmidic replication. The zero-blunt topo PCR cloning kit from Thermo Fisher has a plasmid with blunt ends, and the topoisomerase is able to ligate the PCR products with blunt ends. The selection method is by insertion in the sequence of a lethal protein CCDB. Thus, recombinant plasmid allow bacteria to survive when non-recombinant plasmid kill bacteria when plated. Directional topo cloning consists of a ligation in a linearized plasmid that has one overhang in the 5' end and a blunt end in the 3' end. Usually is a 4-nucleotide overhang, with the sequence GTGG. It requires that the blunt end of the 5' in the PCR product to have the complementary CACC. Ligation can be verified with an enzymatic restriction to be read in an electrophoresis gel, blue-white screening, or the blunt end selection of a fusion-lethal protein interruption.
The coloration of transformed cells is not always reliable. Sometimes the Petri boxes present satellite colonies around the resistant cells. These colonies, which are white, take advantage of the resistant halo formed by the cells with the ligated plasmid. They originally do not have the new gene in their genome, but the expression of blue color is not necessary thanks to the resistant halo in which they are. The efficiency transformation can change according to the state of the plasmid inserted in the cells. For example, if it is supercoiled, the cells have no big trouble inserting the plasmid in its genome because it is smaller and easier to introduce through the membrane than if it is relaxed or lineal. In these states, the molecule is bigger and harder to introduce. The cells used for ligation are the top 10 cells, which have some important characteristics of this process. Its genotype is the following. Some of the most important elements in its genotype are lag-zeta delta M15 element required for the beta-galactosidase complementation when plated on X-gal. Ay, perdón, repito eso, se me sale el Spanglish. Some of the most important elements in its genotype are lag-zeta delta M15 is an element required for beta-galactosidase complementation when plated on X-gal. The GRAS A96, which is a DNA GRAS mutant that produces resistance to nalidixic acid. The genotypes MCR, BC, and MRR, which are mutations that allow methylated DNA to not be recognized as foreign. They are necessary when cloning genomic DNA or complement DNA. The GALK genotype, which is a galactokinase mutation for blocking catabolism of galactose. And finally, the F genotype, which is a self-transmissible low-copy plasmid used for the generation of single-stranded DNA when infected with M13 phage. As it was expected, the efficiency of this ligation was not very high, but still acceptable. In order to improve this efficiency, it is necessary to respect the recovery time for competent cells according to the use method. To choose correctly a transformation method depending on the transformation objective, and finally, have a good cell efficiency since the beginning. Ay, perdón, ya es súper tarde. Perdón si me trabé en algunas partes.